Hello, my name is Professor Hudson, and you are about to view narrated PowerPoint lectures for Module 7, Part 2. In this module, we will talk about rules of inheritance that do not follow Mendelian rules of inheritance. For Module 7, Part 2, the topics to be covered include Section 11.5, do Mendelian rules apply to the inheritance of all traits? 11.6, how are genes located on the same chromosome inherited? And 11.7, how are sex and sex-linked traits inherited? Let us begin. In Module 7, Part 1, we discussed inheritance patterns that follow Mendel's rules of inheritance. Under Mendel's rules of inheritance, each trait is encoded by one gene, and that one gene has two alternative versions called alleles. Under Mendel's rules of inheritance, one allele is always clearly and completely dominant over the other allele. Now we will discuss inheritance patterns that deviate from Mendel's rules. On this slide, we have several examples of inheritance patterns that do not follow Mendel's rules. On the subsequent slides, I will show you some examples of these. So we have incomplete dominance, multiple alleles, codominance, polygenic inheritance, pleiotropy, environmental influence, and incomplete penetrance. Let's start talking about each one of these in more detail. Incomplete dominance. In incomplete dominance, the phenotype of the heterozygote is intermediate between the phenotypes of the homozygotes. Basically, in incomplete dominance, the heterozygous will have a phenotype that looks like a blend of the two parents. Normally, with heterozygotes, whatever is linked to the dominant is what would show up in the phenotype. However, in incomplete dominance, this does not occur. Human hair texture is an example of a trait that follows incomplete dominance. Human hair texture is influenced by a gene with two incompletely dominant alleles. In this example, we will call them H1 and H2. A person with two copies of H1 will have curly hair, as in this example. Someone with two copies of H2 will have straight hair in this example. However, a heterozygote which inherited the H1 and the H2 will have hair that's in between curly and straight, which will be called wavy hair. Let us review. So incomplete dominance is characterized by inheritance where neither allele is dominant or recessive. The heterozygote will be a blend of the two parents. Here's another example of incomplete dominance. Flower color in snapdragons is inherited by incomplete dominance. If you cross a red flower with a white flower, you get offspring that are pink. The heterozygous, or the flowers that inherit a red allele and a white allele from their parents, end up being pink instead of being red or white. The pink color represents the heterozygote phenotype. Note that in the case of incomplete dominance, the way we write the genotypes is a little bit different from the way that we write them under normal dominant recessive rules. Under incomplete dominance, sometimes we use two different letters to represent the different versions of the alleles. In this example, R represented the red allele and W represented the white allele. And then if an individual inherited a red and a white allele, their phenotype would be pink. This slide is also showing incomplete dominance in snapdragons, and it's showing how if you take two pink flowers, which is the heterozygote, and mate them together, you actually can get offspring that are red, pink, or white. Remember, the pink condition is a heterozygote, so they are carrying the allele for the red and the white, which is why you can have red, white, or pink offspring when mating two pink snapdragon flowers. Now let's talk about multiple alleles. And a good example of multiple alleles is illustrated by the ABO blood type grouping. Under multiple alleles, a single gene can have more than two alleles. This is different from what Mendel determined. An individual may have at most two different gene alleles. However, a species may have multiple alleles for a given characteristic. So while you may only have two alleles for a particular trait, in the population there could be more than those two. And depending on who you mate with, your two alleles are going to can be passed on to your children, along with a combination of what other alleles are in the other parent. With blood typing in humans, there are 4 different blood types. They're called type A, type B, type O, and type AB. There are 4 different blood types and 3 different alleles. So remember, Mendel stated that there only are 2 alleles for a given trait. However, we now know that while an individual may only have 2 alleles, there can be more than 2 in the population. When we talk about blood groups and blood group genes, be aware that the A allele and the B allele code for enzymes that add different sugar molecules to the ends of the glycoproteins that protrude from the red blood cells. And the O allele codes for a non-functional enzyme that doesn't add any sugar molecules. So the A allele encodes for an enzyme that will make A sugars. The B allele encodes for an enzyme that will make the type B sugars, and the O allele encodes for a non-functional enzyme that doesn't make any sugars. The blood types A, B, and AB and O arise as a result of the actions of these 3 different alleles: the A allele, the B allele, and the O allele. This slide is just showing the different ways in which the phenotypes, which is the actual blood type that you have, the different ways in which geneticists will write the genotypes for these blood types. So for example, the phenotype type A can be written in terms of its genotype as AA, IAIA, AO, or IAI. The first 2, where there are 2 A's, represents homozygous condition for type A. And the AO or the IAI represent being a heterozygote for the type A condition. Type B can be written similarly. Type AB is written IAIB, or sometimes people will just write AB. And then type O is usually written with 2 lowercase i's to signify that the person neither received an A allele or a B allele. Let's talk about another situation that didn't follow Mendelian rules of inheritance. Let's talk about codominance. In codominance, neither allele is traditionally dominant or recessive. And in the case of codominance, both alleles will try to show up simultaneously. So for example, if you crossed, let's say you crossed a purple flower with a white flower under codominance. If codominance was at play, you would get a plant that had both purple and white petals in the same plant. So in the case of codominance, the phenotypes are not a blend. Both phenotypes show up at the same time. Blood types also illustrate codominance. In the case of blood typing, if you are type AB, you are expressing the A antigen and the B antigen at the same time on your red blood cells. Neither one is completely dominant over the other. This slide is just showing a little information on how blood types work. In that, for example, if you're type A, you're going to have the type A antigen on your surface of your red blood cell, which means you will make antibodies against type B. So if you're type A, you cannot have type B blood or type AB blood. However, you can receive A blood or type O blood. If you are blood type B, you will have B antigens on your surface of your red blood cells, but you will make antibodies against the type A antigen. So if you're blood type B, you cannot receive type A blood or type AB blood. However, you can receive type B or type O blood. If you are blood type AB, you have both A and B blood antigens on the surface of your red blood cells, so you will produce no antibodies to A or B, which means that if you're type AB, you can receive blood from type A people, type B people, type AB people, or type O people. If you are blood type AB, you are considered a universal receiver. You can receive blood from any of these 4 blood types. If you are type O, that means you lack both A and B antigens. That means you will produce antibodies against type A and type B. If you are blood type O, you cannot receive type A, you cannot receive type B, and you cannot receive type AB. If you are type O, the only blood type you would be able to receive is type O. However, you are considered a universal donor if you're type O, because you can donate to any of the other groups. Blood types is an example of multiple alleles and codominance. On this slide, we have an example of how you would do a Punnett square with blood types. It says, draw a Punnett square showing all of the possible genotypes for the offspring produced by a homozygous type A mother and a type AB father. So what are we crossing here? So we're crossing a type A homozygous mom, so we'll write that as IAIA, with a type AB father, so we'll write that we'll write that IAIB. And let's take a look how the Punnett square would be set up. So this is how the Punnett square would be set up. Note in this example, if you cross a homozygous for the A allele type A mother with a type AB father, you will get children that are either type A, 50% of them have a chance of being type A, and 50% have a chance of being type AB. Polygenic traits are traits controlled by 2 or more genes, and each gene will have 2 alleles. Traits inherited by polygenic inheritance will show a wide range of phenotypes. They can be continuous or discrete and defined. Skin color is an example of a trait inherited by polygenic inheritance. Note that in this case, there are actually 3 genes and 6 alleles controlling the inheritance. This is why you get such a wide range of skin colors. Skin color is also an example of incomplete dominance in that if you inherit some of the very light alleles and the very dark alleles, you will have a medium brown complexion. Height is another example of a trait inherited by polygenic inheritance. Traits affected by polygenic inheritance are often strongly affected by the environment, which further blurs the differences among the phenotypes.